hope you people could take a little break. And so I will continue on the theme that we've been talking about using optical tweezers. But now the focus will be on some of the work that we actually do in the lab. Can you hear me clearly? Hello?
Yes, Roop.
Yes.
Okay, good. So, right. So I work at the Indian Institute of Technology, Bombay, which is in the western part of India. And these are the people in my lab. My lab recently moved from the Tata Institute of Fundamental Research to IIT Bombay. And most of the people who you see here are from my old lab, but they are hanging on for about a bit as I collect newer people. So, I mean, I told you about what optical tweezers are, and Avin showed you, really beautiful examples of how you can do very precise single molecule experiments using optical tweezers, and you can combine microfluidics and different kinds of microscopy with the force measurement. And then there was a question at the end of this that how do you, whatever you learn from these kinds of so-called artificial looking experiments, how do you correlate with real things in biology, with physiology? And that is something which is very close to my heart. And I mean, if you ask me, one purpose is of my lab, it is to try and use these kinds of biophysical, seemingly more technical assays to really understand dirty biology. Okay. Now, that may put many people off because, but that is really what is exciting to me, to see how these molecules and these processes act in inside cells, to try and understand this using these kinds of techniques, and to then take it not just from the level of single organelles and single cells, but to the level of how an animal responds is what really my interest is. And in that context, we try to understand how lipid membranes and these motor proteins that we talked about, they are how they are married to each other and what they do together and what they don't do together. Just to kind of, this, I think this has been repeated quite a few times, but we are interested, if I ran this movie, what you would see here is a kinesin motor that we showed, carrying a vesicle. Let's say this could be a mitochondria, this could be different kinds, this could even be a small virus or a bacteria inside cells, and it is walking along these microtubules. And what this kind of motion does is that it takes things to the right places inside cells. And now you have to, so there has been a lot of focus in understanding these kinesin motors and how they step, etc., etc. But what has mostly been ignored is this lipid membrane, this blue thing that you see here, to which these motors are attached. And that is where we kind of have been trying to contribute. And we think it is really important to understand what this membrane does to this motor and vice versa. And there are many aspects to it. I won't be able to go into all these details. But to kind of continue on that, almost everything where a motor generates force inside a cell, it generates actually force on a lipid membrane first, okay? And that lipid membrane may contain a mitochondria, it may contain bacteria, it may contain some kind of a virus, different kinds of particles on which you have these motors. So what does the membrane mean for these motors and vice versa? To try and understand this, so one kind of essays which we discussed was where you take these kind of artificial cargos and coat them with motors which are kind of artificial, and then you look at what happens under fairly high resolution. We can do that kind of work. We have done that kind of work earlier, but now we have moved on gradually, kind of in our own niche, where we try to understand how complexes, large complexes of these motors, how multiple motors, they generate force cooperatively, and what that means for something inside the cell. Now, we do that in different ways. And one of the ways we do that is that we grow cells in a petri dish. Let's say here are cells. And what we do is that we take plastic beads, 1 micron plastic beads, let us say in this case, and feed them to the cells. These cells think of these beads as breakfast, and they will eat it, okay? Because as you know, many cells in your bodies, let's say the immune cells, they are eating up bacteria all the time, and so they will eat these beads merrily, okay? And once these beads get inside, they are not beads anymore. They now acquire, they are not like this, what the typical thing that you have been seeing in our earlier slides. Instead of this bead, you have an actual cellular organelle with many motors and non-motor proteins on this. It has a lipid membrane around this, and this is an example here, okay? These are these beads which are inside this cell, and you are seeing a single cell here. So each of these beads is about 2 microns. So you can get an idea of the size scale here, and you can see that they vigorously move inside these cells, okay? Now what we can do is that we can shoot a laser through this cell and trap this one here, let us say, okay? And see how it moves around, and that we have done in the past, and we do that right now also. But what we can also do is, once these beads, now they are phagosomes. What does that mean? They are beads that have been ingested by cells and they have become actual cellular organelles. We can extract these phagosomes. From a phagosome is a bead which went inside a cell and matured in a certain way to acquire a lipid membrane and motor proteins in a way which is very natural, okay? So we can extract these from the cells, and now we can study these in an optical trap to learn things that we could not have learned by the typical assays where you have a purified motor and a purified bead, okay? So what can you learn? So instead of this, now you have something like this, okay? Hold on. Right, you have something like this, okay? Just hold on a minute, there's something wrong with this. Sorry about that. Right, so can you see my cursor?
Yes.
Right, so you have, let's say this is a motor protein, let's say the kinesin, but essentially if I draw it as a cartoon, you have a situation where you have, let's say, a mitochondria which has been extracted from the cells, and on this mitochondria there are likely hundreds of proteins on a lipid membrane, and some of these proteins are these motor proteins. And in this case, this is of course a very cartoonish view. You can see that there are multiple motors, these dynein motors and these kinesin motors. Typically, they exert force in opposite directions. The kinesins want to go towards the plus end of the microtubule, which is typically towards the periphery of the cell, and the dyneins want to go the other way, which is towards the nucleus, towards the minus end of the microtubule. And this system, how it will behave cooperatively, okay, you can imagine that there are these 2 tiny teams of motors applying forces in opposite directions, and the net balance of this force will determine where this mitochondria will end up. In fact, indeed, it will determine how a cell is organized to a large extent, about depending on how these 2 opposing motors generate force and what their relative magnitudes are. So what you can now do is that you can actually trap single phagosomes, which are not single, they don't have a single motor anymore. They have multiple motors like this, and their lipid membranes and everything else inside it. You can trap it in this optical trap, and you can do the same kind of experiments that we have been discussing. Avin has showed you, and I have also showed you, and you get the similar kind of data. But now this data is much richer, in the sense that the forces are generated by not one, but by multiple motors, and by multiple motors, sometimes motors going in the one direction, at other times in the other direction. And you can really analyze and understand this data and do fairly complicated statistical mechanics on this, but I'm not going to get into that today, okay? Right, so I'm going to talk to you about some very recent things that we have been doing in our lab, try and address a few questions, okay? I showed you this, cargo. Let's say it is that bead which went into the cell and matured to become this phagosome, and it had these opposing motors pulling it. Now, let us forget about the kinesin motors, the green ones, for a minute. You have these dynein motors shown here, and dynein does not come alone. Okay, it comes with a partner almost always inside cells, and the partner here is dyneactin, dynein associated protein. Okay, dyneactin. So here is dynein. It's walking along this green thing, which is this microtubule here, and you have to imagine dyneactin, which is like basically it skates along on this. Okay, and if there is a problem with dyneactin, there are many mutations in this dyneactin molecule which lead to severe neurological disorders. Okay, these are essential proteins. They are important for almost every conceivable biological process that you can think of. Okay, any problem in this, a single point mutation here or here, can have very severe consequences on health. And one of the first places where you see that problem is in neurological disorders, problems with cognition, for example, Alzheimer's disease, and perhaps a simple reason for that is that the neurons are really long cells. Okay, and any problem with transport will get amplified on a long road, obviously. Okay, so there has been a long-standing question about what does this dyneactin molecule, how does it help dynein? Because so now what is interesting is that dyneactin has one attachment to this microtubule. So dynein, so imagine yourself walking on this microtubule, right? Your feet will have to attach to the microtubule, and you will do something like this. So you move in particular direction. In addition, dyneactin also has domains which attaches to the microtubule. Why is this needed? That's not very clear. Okay, what happens if I disable this interaction? What happens if I disable this interaction? Typically, when you want to understand this, the kind of things people have done is that they have overexpressed, expressed extra copies of this one, or some domain of this inside cells, and try to see coarsely what happens. Now that is fine, but it's very hard to interpret those experiments because you don't know what is cause and what is effect. On the other hand, you can purify this dyneactin, you can purify dynein, and separately, bring them together on the surface of a bead and do the kind of experiments that we were talking about to try and understand things. That is great, and a lot of people have done that, but those experiments become too artificial. You do not know whether these are combining in the right way, and there is no membrane on it, and there are no multiple, there are individual motors. So these also, there are problems with this. So we wanted to be somewhere in the middle of this, where we extract these native-like cargoes, these guys, these beads which have gone into cells and have all these motors along with the membrane on it, and extract them and look at them in this optical trap. Okay, when we do that, now if we want to understand, I'm just trying to give you an idea of the way in which you do these experiments. I want to understand what the interaction of dyneactin with dynein does here. Okay, so what I can do is that I can take a small peptide. I hope many of you know what a peptide means. It's a small part of a protein. Okay, what is that protein? This peptide is the part of dynein that binds to dyneactin. Okay, so now let's say I put in 100 such peptides into this solution. That part, this small guy, will go and bind to dyneactin, right? It will mimic this large molecule, that this small part of the large molecule will go and bind to dyneactin, and what it will do is that it will cause dynein to separate out from dyneactin, right? I hope everybody understands this. If you don't, please ask me. Okay, now, so by doing this kind of an experiment, you can specifically ask why this interaction between dyneactin and dynein is needed. So when we do this experiment, so we basically have these beads which we have taken in phagosomes, which we have taken inside cells and purified, and we measure the force generation of dynein with or without this peptide added, and see how do things change. Okay, so without going to the details, there's a lot of background work to this which I won't go into right now. You can at a very first order level, you can measure two things. I guess all of you understand what these plots mean. Okay, I will repeat. This is time and this is distance, which you can multiply by the trap stiffness to get a force, and you have to imagine that this is what is happening. Okay, here is dynein, dynaactin pulling this phagosome out of the trap, and then it keeps falling back and generates. Now you can simply count the number of, so this is one force generation event, this is another one, and this is another one. And let us say within a time duration of 4 seconds, you had 3 events here. So you can simply, this is a very nice way to measure the on rate of this molecule, right? If you count three such events in 4 seconds, then you can get an on rate directly. This is something which is typically measured by biochemical experiments, by doing complicated biochemistry, but here is a direct measurement of an on rate of a motor working in its native-like condition. And if you measure that once it starts generating force, how long does it stay attached, and when it detaches, if you measure this duration, that gives you the off rate, right? If the duration is longer, then the off rate is lower. So now we wanted to see that now what we observed is that if you add this peptide, then the on rate, the number of events per unit time, are reduced by this peptide. However, the time for which these events are generated, that is the persistence of this motor, the tenacity of this motor against opposing force from the optical trap. You have to appreciate this, that this duration tells you the tenacity, the persistence of this motor against opposing force from the optical trap, right? And the duration of this event, if you take 1 by that duration, that is off rate, right? 1 by this duration is the off rate simply. There was no effect of this peptide, dynein intermediate chain. So in other words, things look a little bit like this, okay? If I break this interaction between this dynaactin and the dynein here, this engine has a starting trouble, okay? It can't start. So it can't attach to the microtubule, and it has starting trouble, K on. Now let's do another experiment. Now let's try to understand why does dynaactin attach to microtubules? Remember here, okay, so the dynaactin attaches to microtubules. This is something that we have already tried to understand here, what I told you. And now what we do, we take an antibody. The antibody is against this region of dynaactin which attaches to the microtubule. This antibody, if it goes and sticks here, it should block the binding of dynaactin to this microtubule. So let us see what happens then. Okay, so now we are doing a similar kind of experiments with an antibody blocking this interaction, and we simply count how many events per unit time and how long. Now what we find very interestingly is a completely opposite effect. The antibody means that blocking this interaction has no effect on the on rate. How many times you will see these kind of events per unit time. And however, the time, I'm sorry, this arrow has moved here, this should be here. How long this whole molecule, the dynein dynaactin complex, can persist against force, that gets reduced drastically by the antibody. Okay, so what that means is the following, that the dynein starts, the complex starts off fine, but as it starts moving, then it has a problem sustaining force. So this is what happens to the engine, that's to the car. Okay, so just to summarize this, this link between dynaactin and dynein is required for this entire motor to engage. Okay, but once it is engaged, it seems to have little role in sustenance of this engagement. For that, you need this dynaactin to engage with the microtubule and stay there. Now, I'm not going to show you the details, but you can actually try to understand this in terms of a mechanical torque force that is being exerted when you are pulling something back. But that's something which, if somebody wants, I can discuss offline. And this paper should appear sometime soon. Okay. If there are any questions related to this part, I can answer now, because I will switch to something completely different in the next few slides.
I have a question.
Please.
I was curious because with your phagosomes, you have multiple
Can you
dyneins.
speak a little louder? I can barely hear you.
Okay, sorry. Can you hear me now?
Yeah, better.
All right. So with your phagosome system, you have multiple dyneins, right?
Yes.
So obviously, when you add the peptide, you would, I mean, it wouldn't bind. But when you haven't added it, wouldn't you? I mean, you have multiple dyneins. You don't really look at just one molecule.
Yes, you're absolutely right.
Yeah. But
yes.
Maybe that isn't an issue.
No. So the on rates and the off rates we are measuring is not of a single dynein. It is of a team of dyneins. Yes, absolutely right. But that is more physiologically relevant.
Yeah.
Yeah.
All right. Thank you.
Okay. Any more questions related to this part? I hope people understood basically what we are trying to do here.
But Roop, just to follow up on Oscar's question,
yes,
have single molecule experiments been done with the dynein interacting protein, the peptide that you use, or the antibody?
Yes, I mean, these single molecule experiments have been done, but nobody has actually used the optical trap to measure, kind of infer these on and off rates.
Clear.
Hello?
Yeah, it's clear.
Yeah, so, right. So people have done optical trapping experiments, obviously, but they have not tried to use the optical trapping data to infer an on rate and an off rate. Right. And neither have they tried to disrupt these interactions in the manner that I described. So, this dynein-dyneactin field is quite complicated, and there's a lot of papers on this.
Yeah.
We can talk about this at some time. It become too technical if we get into that right now. Okay, so what we can also do is, imagine this. Okay, this you see on Indian roads quite often, and imagine a microtubule like this. Okay. And an obstacle on this. And this is really important because the inside the cell is an extremely dense place. We have the same microtubule is being used for many other cargoes, many other motors. So the question is that if the dyneactin microtubule interaction, what I showed you here, this is required for sustenance, okay, for sustained force generation, it is very likely that when there is a mutation here, then you will not be able to move in a crowded environment, okay, where you have to generate sustained force. And this is actually something that has been kind of inferred by people. People suspect this because this is important for understanding neurological disorders, where inside the narrow axon, if you have to move, it's not going to be easy to move if you have a mutation here. Okay, so what dyneactin might be doing, it keeps the system attached so that it can generate persistent force and move through narrow pathways or against obstacles. That's something we tried to kind of get from using our optical trap data, basically trying to use the optical trap, the force from the optical trap, to simulate a narrow axon or an obstacle. Okay, but we are trying to do this in different ways, where you have such a phagosome and you put it along a microtubule and you place an obstacle on the microtubule. Okay, and see what happens on the obstacle if you prevent dyneactin from attaching to the microtubule. So I feel that we can really learn new things which are important to health and disease by doing these kind of experiments, biophysical experiments also. Now I'm going to switch to something completely different. Okay, and so here is what I have been talking about. Here is, let us say, one of those phagosomes, or it could be a bacteria, it could be a mitochondria, where basically you have some internal stuff which is enclosed in a bilayer lipid membrane, and there are motors attached to this. Now, we very casually, whenever we talk about this, we talk about a bilayer membrane, but it need not be so always. Okay, all this stuff that you eat, okay, all the fat that you eat goes and sits inside organelles, inside almost all kinds of cells in your body. And those organelles are called lipid droplets. And these lipid droplets, because the internal matter is fatty, hydrophobic, they have a monolayer phospholipid membrane. I guess if you think about it, it's obvious that if you have water inside, you will have a bilayer membrane because you have charged phosphate groups here. But if you have fat inside, then you will have a monolayer membrane because this fatty acid tail is hydrophobic and it's happy with this fat here. Okay. Now, whatever was happening on a bilayer membrane need not be true on this monolayer membrane. The proteins that attach to the monolayer membrane are likely to be very different from the ones that attach to the bilayer membrane. And this brings in really interesting physics, chemistry, and biology into this. Okay, how do these membranes fluctuate? What is the organization of this monolayer membrane? Really, nobody knows that. So that's something which we are very interested in. Particularly, we are interested in understanding how the attachment of these motors to these monolayer membranes is relevant for a host of metabolic diseases, including liver disease, obesity, and so let me tell you the reason for that. What you see here are a few cells. So there are two cells where these lipid droplets, these are almost perfectly spherical objects. Why? Because they are oil in water. Okay, so they want to reduce the surface area, they are going to be spherical. So they are excellent for optical trapping. All right. So these spherical lipid droplets, they are stained with some fluorescent dye, and you can see them move around a lot, okay, inside these cells. And the reason they are moving around is because they have these kind of motor proteins on their surface. Why are they moving? Nobody really knows. Okay. We think from some work that we have done very recently is that this motion of these lipid droplets allows this fat to be exchanged with other organelles and to be sent out from cells. And what we showed is that very recently is that this is really important for how your liver functions. Okay, so what we were able to show, and this I'm just really going to skip over all this, is you go through your feeding fasting cycles every day. Okay, now you would probably not survive even one feeding fed fasted cycle which you go through every day if your liver was not managing the fat in your body. What happens is that your adipose tissues, your fatty tissues, when you don't eat, they send out a huge amount of fat into your blood. Okay, that fat goes to your liver, and the liver is like a sponge which keeps this fat, because otherwise if this fat went into your blood and it went to your heart, it would probably kill you within a few days. Okay, you know what atherosclerosis is, basically accumulation of fat in your heart. And what we were able to show is that kinesin motors here attached to a specific lipid called phosphatidic acid I don't want to go into the biochemical details of this, of lipid droplets inside the cells of your liver. We could show this inside the liver actually, that these lipid droplets are then transported to the periphery of these hepatocytes, which are the cells of the liver, and then they are secreted out into blood so that you can, and this secretion changes across your feeding fasting cycles every day so that you can maintain a constant level of fat in your blood irrespective of whether you are in the fed state or the fasted state. This entire pathway we were able to show is controlled by insulin, and that's really fantastic because insulin is obviously the one hormone that comes to your mind when you talk about fed fasted cycles, right? So I won't get into this too much. Let me skip this. What we are doing right now is, in many contexts, whenever chemistry has to happen inside cells, components need to mix. Okay, here is one organelle, let's say mitochondria, something is contained inside this. Here is another organelle called, let's say, a lysosome. These two have to mix. So these membranes must somehow fuse transiently, and then things will mix, and then something new will happen, right? And one of the most extensive membranes that you will see inside cells is the endoplasmic reticulum. Okay, so the endoplasmic reticulum is stretches everywhere, and it exchanges lipids and proteins with almost every conceivable organelle inside the cell. peroxisomes, endosomes, lipid droplets, the one that I talked to you about, ER, and this has been studied for a long time. We wondered if these contacts, so these contacts between, let's say, mitochondria and ER is essential for, almost, so many kinds of different processes which are important for metabolic, metabolic diseases as well. So what we wondered, if we can reconstitute these interactions on a cover slip, okay? And so what we did, and this is something that we are right now trying to, write up, is that we purify endoplasmic reticulum in the form of small liposomes from the liver of a rat, okay? And this rat can either be in the fed state or in the fasted state, or a rat for where we have activated its immune system by giving it some kind of a bacterial load, okay? And now we use an optical trap to understand whether the membrane of this endoplasmic reticulum interacts differently with other organelles, okay? So what do? We basically prepare something called microsomes, okay? Basically, purify an endoplasmic reticulum fraction by centrifugation from the liver of a rat. And now this comes in the form of these small liposomes, and I won't go into the details of this. You can actually deposit this membrane on a cover slip, and we are able to show that this is a smooth bilayer membrane with many of the proteins that were present on this endoplasmic reticulum, they're actually deposited on this membrane, okay? Now we want to understand whether the lipid droplets, so remember what I showed you in the previous slide here, that these lipid droplets interact with endoplasmic reticulum. Now this endoplasmic reticulum, this complicated thing inside the cell which is hard to visualize, I have been able to stick it to a cover slip as a planar membrane, okay, as a mimic of the endoplasmic reticulum. Now I flow in lipid droplets from the top, okay, and here are lipid droplets, and I flow them into a flow cell, and then I let them be for some time, and then I wash it, okay, and a certain number remains, and I can visualize this very nicely, okay. So the difference, the number here divided by the number here, pre-wash divided by post-wash, gives me a very nice estimate of the ability of lipid droplets to stick to the endoplasmic reticulum, okay. What I can, so what I see is that if I take lipid droplets from a fed animal, many of them remain stuck even after I wash. If I take lipid droplets from a fasted animal, they don't stick. If I take lipid droplets from an animal where I have activated its immune system by providing it lipopolysaccharide, okay, they start sticking. Now we are also able to find out the proteins which are responsible for this sticking. What more than that, because this is a talk on optical tweezers, what we can do, we can use an optical tweezer to find out what are the proteins which are responsible for this tethering. So what we simply do, we have this membrane. It is mimicking the endoplasmic reticulum on this cover slip. We call it the microsomal supported lipid bilayer. Imagine that ER to be here. We trap a lipid droplet or a bead in an optical trap and bring it and lower it onto this. Then, so this is here, a bead, and we lower it here. And then we simply move the underlying endoplasmic reticulum or mimic microsomal supported lipid bilayer at a constant velocity, okay? If the lipid droplet interacts with this, so that's what we are moving this, okay? Move with the piezo state. And if there are proteins which interact with this, the lipid droplet or the bead will get stuck to it, right? That's exactly what you see here. If I just take a simple bead and this is time, if I move the underlying, this one, nothing happens. The bead stays wherever it is because it is trapped in the optical trap. But if I take a bead that is coated with kinesin, okay, you see that it will get stuck and it will move out. This does not happen with dynein, and we have done quite a few other controls which I'm not going to talk about. But basically, you can reconstitute membrane-membrane contacts in a metabolically relevant and immunologically relevant manner on a cover slip. And we are going to have quite a lot of fun with this, I'm quite sure. So, in other words, you know, yes, an optical trap, whenever you hear about it, you think about measuring force, you think about doing very high resolution experiments. Those are fantastic. But you can do much more simpler things, okay, with an optical trap. You can engineer events. You can engineer membrane-membrane interactions and understand a lot of things which are otherwise not possible to understand inside cells. I think I will stop there. I would rather take more questions than go on with my talk. This is where I am. This is the view from our rooftop. That's Powai Lake in Bombay. And we are funded. We are very grateful to these IIT Bombay and the Welcome DBT India Alliance for funding us. If people are interested in postdoctoral positions where you do physics, but you do chemistry, but the purpose is to learn biology, then please write to me.